Hey everyone, this is video number 1 for module 7, and this video is, this module is about gene regulation. So I've recorded a number of short videos with kind of whiteboard definitions and outline of the main topics that we'll cover. And in future videos, I will talk about specific examples from both prokaryotes and from eukaryotes. All right, module 7 is about gene regulation. So I wanted to start with an introduction to the topic, and then we'll go through some specific examples that illustrate how genes are regulated. So first of all, I just want to mention some main points that we'll be covering, and I'm just going to run through these in this video. So first point is organisms are equipped with more genes than they need at any given time or place. So it's not the case that all the genes that an organism carries are always on. So some of them at any given time and place are on. The majority usually are off. So some genes are going to produce proteins that are going to be needed in certain situations. So the idea of gene regulation occurs throughout biology, all organisms. It's the norm, and it's happening to most genes. They're in either in an on state and expressed, or an off state and not expressed. And they can switch between those two, from off to on or on to off. Okay, so expression is regulated, that's the norm, and genes are expressed according to different timing situations. So, for example, organisms that undergo development will turn on a series of genes that will allow the different processes involved in development to be carried out. And normal development requires a very elaborate set of genes at a certain specific timing in order to work properly. Expression can be regulated in different conditions in different places. So, for example, plants turn on genes in sunlight or in dark. I guess that's not really a place, it's more kind of an environmental condition. And genes can be used in different processes. So sometimes if that process is being carried out, for example, production of gametes, there's a whole set of genes that is necessary for that. Or dealing with toxins in the environment, detoxification, right? All right, so expression is basically, you can think about it as meeting the needs of the organism. And what I mean by regulation is when a gene is on, it's being expressed. It's going through the process of transcription, translation, making a protein that's active to carry out its function. When a gene is off, there's something interfering with that whole process such that no active protein results. Okay? Second point, and this is going to be a preview of how this module is broken down. Think about regulation in organisms that are unicellular, and I'm going to take this to mean prokaryotes versus multicellular, and I'm going to take this to mean eukaryotes. I know there are exceptions. There are unicellular eukaryotes and there are multicellular prokaryotes, but for now I'm just going to equate the two, just for simplicity. An organism that's unicellular is in direct contact with its environment. And so gene regulation in unicellular organisms is usually about being able to take advantage of or be resistant to certain environmental conditions. So, for example, unicellular organisms in very cold environments make an antifreeze protein. When the environment warms up, there's no longer the need for that. So that's an example of a gene that's regulated in response to a changing environmental condition. Also, in dry environments, they can undergo what's called sporulation, formation of a tough coat that allows them to survive the desiccating or dry environment. Regulation in the environment can be used to take advantage of nutrients that an organism might find in its environment. So in some situations, there may be simple sugars that are available. They can take those in, break them down to make ATP. Or they might find them in another environment where there are a lot of lipids, okay? So in order to take advantage of those conditions, they have to alter how they take things in and how they break them down. So when we talk about gene regulation in prokaryotes, which are mostly unicellular, we're going to be talking about that, how genes regulate in response to mostly nutrients are the examples we're going to talk about. Availability of nutrients, how does gene regulation work? Think about the situation in multicellular organisms. So multicellular organisms are more insulated from their environment. Sure, they interact with that, but the way multicellular organisms work is they create a really large body, and different cells within the body are specialized to carry out different functions. So gene regulation, not entirely about different types of cells, but that's sort of the context in which it's been studied in multicellular eukaryotes. That is, how do we, starting as a single cell, a fertilized egg, go through the process of rapid cell division and develop an organism, an adult organism, that has many different cell types, cell differentiation, that are specialized for different functions. So that's sort of the context that we'll talk about gene regulation in multicellular eukaryotes. How does a nerve cell get supplied with a set of proteins that are going to carry out conduction of nerve impulses? And how does a muscle cell get supplied with proteins that are going to carry out contraction of the muscle? All right, so those are 2 points to consider. Okay, 2 more points. Point number 3. Regulation can occur at different levels of gene expression. So gene expression, DNA is transcribed to RNA, which is translated to protein, and then the protein has some activity, that's resulting from all this process. Regulation can occur at all of these steps: initiation of transcription, the process of transcription itself, termination of transcription, stability of the RNA, initiation of translation, progression of translation, termination of translation, stability of protein, and even modification of protein. So it's a huge topic, and I'm going to choose one part of that. The most commonly studied part of regulation of gene expression, and the most common point at which regulation occurs, is at the very beginning of the process, the initiation of transcription. If you think about it, energetically, it saves energy to regulate at the very beginning of the process. If you don't need a protein, it consumes energy to go through transcription and translation, and only regulate it at the end. So this is the most common mechanism of regulation, transcription initiation. Okay, so point 4 here is mostly a reminder about what's happening in initiation of transcription. We covered this way back in the beginning of the course. And here's DNA, double helix. It's going to be a gene that's going to be transcribed. There's a start site of transcription. This is where the RNA polymerase is going to start making an RNA copy of the DNA template. In order for RNA polymerase to find the place to start transcription, each gene is equipped with a consensus sequence, or sequences in some cases, right next to the transcription initiation site. That's called the promoter. So the first thing that must happen is that the RNA polymerase, this green blob here, must bind to the promoter. That's it, puts it in position to then start making the RNA product strand from the DNA template. So just some terminology. We're going to talk about positive regulation and negative regulation. If you're talking about transcription initiation, positive regulation is anything that's promoting RNA polymerase activity. That could be binding to the promoter or the rate at which it makes the RNA product. Anything increasing that rate or making binding more likely is positive regulation. That's going to increase the rate of RNA product synthesis, positive regulation. Negative regulation is anything that interferes with that or slows it down. So there are some things that interfere with RNA polymerase binding to a promoter. That's a form of negative regulation. Okay, so that's 2 more points. All right, point number 5. Now we're going to restrict this to specifically talking about gene regulation in prokaryotes and at the level of transcription initiation. Okay, we're kind of zeroing in on our main focus. So in prokaryotes, there are 2 main mechanisms regulating transcription initiation. One of those mechanisms is called inducible regulation. Inducible regulation occurs for genes that encode enzymes that are catabolic enzymes. Catabolic enzymes, enzymes that do breakdown or hydrolysis. So it's breaking down a large molecule into a smaller product molecules, okay? In inducible regulation, the gene is usually off, but it gets switched on, meaning a functional protein or proteins is produced when the catabolite is present, all right? So these genes are only expressed when some catabolite is present in their environment. And remember, for prokaryotes, they're in direct contact with their environment, okay? So an example of that is in the bacterium E. coli. When you drop E. coli into a solution or, let's say, water containing lactose. Lactose is a disaccharide. The E. coli will produce a catabolic enzyme, induce that gene to express that enzyme when lactose is present. And that catabolic enzyme has the function of breaking lactose down to its component monosaccharides, galactose and glucose, okay? So that is inducible regulation for genes encoding catabolic enzymes. Okay, the second main mechanism by which genes in prokaryotes are regulated through transcription initiation. Transcription initiation in prokaryotes is called repressible regulation. Repressible regulation is for genes encoding anabolic enzymes. In repressible regulation, the gene is usually on, but is switched off or repressed when a biosynthetic pathway end product is abundant, okay? And just to remind you, anabolic enzymes are enzymes that carry out synthesis reactions, taking smaller substrates and building larger products. An example of that is seen in E. coli, and this is the biosynthetic pathway. I haven't given the name of the intermediates here, but it's a pathway in which the end product is the amino acid tryptophan. The enzymes that are catalyzing these steps are anabolic enzymes. And these enzymes are regulated through repressible regulation. So we want them to be on in order to make this amino acid if the E. coli cell needs it. However, if tryptophan is present in abundance, there's no need to make more. So these enzymes are shut down, the pathway is shut down, it is repressed by repressible regulation, okay? That's the second main mechanism. So inducible regulation is for enzymes that are encoded, that are catabolic breakdown. Repressible regulation is for enzymes that are anabolic synthesis, okay? All right, the last point I want to make is just a reminder about the structure of prokaryotic genes. There's a unique feature of that. So in prokaryotic genes, the proteins that have a common kind of function are grouped together into a structure called an operon. And the operon is under common regulation. And so these genes are expressed together, usually, almost always, because whatever's regulating their expression is going to cause them all to either be expressed or all to be shut off. So the operon looks something like this. At the DNA level, the blue line here, there is a promoter which is going to be guiding RNA polymerase to bind and to the site of transcription start. Transcription will start here and make an RNA, and it's going to transcribe however many genes are in the operon. This example has 3 genes. In the DNA level, the genes are separate coding segments. The RNA transcription will transcribe one transcript, one mRNA, that will have 3 different coding regions, okay? This is called a polycistronic mRNA, meaning it has more than one cistron or protein coding region, okay? This mRNA is going to undergo translation, but for each gene, translation is separate and independent. Transcription is common, it's under common regulation, but when translation occurs, the ribosome is going to attach here and translate this and stop. Another ribosome here, translate that and stop. Third, there. So you get separate proteins being produced from a common mRNA, okay? So we'll look at examples of 2 different operons. One of them is an example of a gene that is encoding enzymes that are catabolic enzymes, and that's an example of an inducible operon. The other example is enzymes that are anabolic, and that's an example of a repressible operon. Okay, so that was an overview of Module 7 and the main kind of definition of factors that are involved. In the next video, we'll talk about the lac operon, which is an example of inducible regulation. See ya.